Hey everyone, my name is Sono Khan, and I'm the director of the Queen's Genetically Engineered Machine, or QGEM team. We are an undergraduate student-run interdisciplinary design team comprised of life sciences, biochemistry, and engineering students. Every October, we represent Queen's University on the world stage at the International Genetically Engineered Machine, or iGEM, competition. Each year, we work on new research projects with the purpose of solving real-world problems using synthetic biology. We mainly operate over the summer, and our findings over the summer are presented at the annual iGEM competition, where we compete with over 330 teams from universities around the world. The iGEM competition has quickly grown into one of the most prestigious international research conferences in the field of synthetic biology, and our award-winning team is looking for new students to come on board and help us continue our successes into the coming years. So over the past few years, QGEM has developed some really interesting projects, and we've done really well at the iGEM competition. In 2018, we developed a luminescent biosensor for hormone detection and diagnosis. We created a pacifier-like device that could be able to quantify levels of cortisol in saliva and therefore monitor stress in kids. And in 2019, we developed a THC biosensor using fluorescent antibodies for roadside THC detection. So the QGEM team is divided up into three main sectors. We have our wet lab team, our dry lab team, and our outreach team. The wet lab and dry lab team will work together to develop this year's project, which is a transdermal biosensor that frequently and accurately measures levels of hormones and minerals very important for chronic kidney disease patients. The dry lab team will also help develop our wiki, which is where we showcase all of our project information. And then the outreach team will work together to cover the social aspects of the project, including human practices, public engagement, and collaboration. I'll now pass it off to Cody and Jen, who will talk a bit more about the wet lab and dry lab side of QGEM.
Thanks, Sono. I'm Cody, and this year I'm the wet lab lead for the QGEM team. I think one thing that really surprised me was just the field of genetic engineering and how we apply certain engineering principles like design, modeling, and testing to fields like synthetic biology. Now, with synthetic biology, we could insert foreign DNA into a cell to grow, let's say, proteins or create other non-native functions. And for example, I guess we could take organisms and we could actually grow them so they produce a green color and they just glow under UV light. Or we even can make bacteria to break down plastic in landfills. And so while obviously there's some ethical concerns, the QGEM team uses our synthetic biology for good, and we get it pre-approved. And this year we created proteins that actually glow up when they bind to certain biomarkers, such as elevated phosphate in patients with chronic kidney disease. So if you join our wet lab, you'll get experience with protein synthesis, DNA design, protein modeling, in vivo and in vitro biological testing, which is testing our product to see if it works, biochemical techniques, and also general research, which would include data analysis and data collection. And I think QGEM is really a great experience. It's amazing that while you can't actually see what really you're creating and what protein you're making with the naked eye, what you can see is the impact that it has and also the impact of synthetic biology.
Thanks, Cody. Now, moving on to maybe a more familiar side of our work for students in Eng, I'm Genevieve and I'm the dry lab team lead for QGEM this year. So the dry lab team members work together with the wet lab to make synthetic biology more usable by engineering an application for it. So this could take the form of creating models of or interpreting data from the wet lab team's work to predict how a device would work, and further working with SolidWorks to 3D print device housings. There's also lots of room for elec and comp in working with Arduinos and circuitry or building software. So in joining our dry lab, you'll learn about synthetic biology far beyond what you would in an engineering classroom, while getting the opportunity to apply your existing and new engineering skills to novel problems.
And that's it. Thank you for watching. If you want to get involved with QGEM, reach out to us at our email, on Facebook, or on Instagram. Thank you. Bye.